AP1 final section 4, the stages of mitosis in order are PMAT, prophase, metaphase, anaphase, and telophase. The DNA code, transcription versus translation. DNA transcribes the code for the cell's activities. RNA translates it into proteins to carry out what was transcribed by DNA. DNA transcribes and RNA translates. A hypotonic solution has a lower solute concentration outside. So water enters the cell swells or bursts, and that's called hydrolysis. So hypotonic is low outside, high inside. So it needs more water inside the cell, but not too much. Hypertonic is a higher solute concentration outside the cell. So if outside the cell is salty, the water flows out of the cell shrivels up, and this is called crenation. Isotonic is where the fluid inside matches outside. It's an equal concentration, so water does not move. Exocytosis is to move out. Endocytosis is where the cell is drinking this interstitial fluid around it. So it's bringing it into. Let's see. Endocytosis - penocytosis. Endocytosis, phagocytosis is the cell is eating solid material like other dead cells. Phagocytes eat the waste. Passive transport. It does not require energy. It follows the concentration gradient, so it moves with the river. It moves substances from high concentration to low concentration. The types of passive transport are simple, facilitated, or osmosis. Simple diffusion or simple passive transport is it moves small or non-polar solutes like oxygen gas or carbon dioxide gas. Facilitated diffusion moves ions through channels. So it moves glucose, which is polar, through a carrier. So I guess that would be like a channel. Passive transport is the osmosis is the movement of water. Active transport does require energy. It's going against the gradient and it also involves the sodium-potassium pump. The sodium-potassium pump moves 3 sodium out and 2 potassium in. Bulk or vesicular transport uses vesicles to move large substances in and out of the cell. The levels of the organelles, I don't know if that's the right term, but no, it's just all the organelles. Mitochondria, smooth ER, rough ER, ribosomes, Golgi apparatus, lysosome, nucleus, nucleolus, peroxisome, cytoskeleton, and centrosome. Mitochondria is the site of ATP production. The smooth ER is where lipids are made. The rough ER is the site of proteins folding into the correct shape Ribosomes are the site of assembly of amino acids into proteins. The Golgi apparatus is the mailroom for proteins from the rough ER. It packages them and sends them out via vesicles to the right spot. A lysosome, it lyses or digests material that has been endosized. It's things that contain the lysosome contains enzymes for hydrolysis reactions. The nucleus contains DNA. The nucleolus is where the ribosomes are made. Peroxisome will detoxify alcohol. The cytoskeleton is the skeleton of the cell. It gives its shape. The centrosome or centriole organizes the cytoskeleton for a normal life cycle. It reorganizes it into mitotic spindles for cell division. Not sure that's spelled right. That's it for 4.